even as those in the third world suffer from malnutrition. Among the leading current causes of disease and death in developed countries are obesity and its corollary, type 2 diabetes. Type 1 or juvenile onset diabetes has long been known to have an autoimmune etiology. Cells of the adaptive immune system kill the beta cells of the islets of Langerhans in the pancreas, leading to the complete absence of insulin in the diabetic patient who must rely on exogenously delivered hormone to survive. In type 2 diabetes, the body fails to respond to insulin in an appropriate manner and the cells fail to take in glucose from the blood and into the tissues. Just as in type 1 diabetes, the presence of high levels of glucose in the blood and the tissue fluids facilitates non-enzymatic glucose conversions to reactive carbohydrate derivatives such as glyoxals. These derivatives cross-link proteins and carbohydrates in the membranes and walls of blood vessels and neurons and within the extracellular matrix, leading to the familiar array of diabetic symptoms, that is, poor peripheral circulation, plaque buildup in the arteries, and heart disease. However, we are now beginning to understand that type 2, just like type 1 diabetes, is a disease closely related to the workings of the immune system. In 1993, a seminal publication by Hatam and colleagues made the link between inflammation and metabolic conditions such as type 2 diabetes and obesity. These authors demonstrated that adipocytes constitutively express the pro-inflammatory cytokine TNF-alpha and that TNF-alpha expression in adipocytes of these obese animals is markedly increased. Later research demonstrated that this finding could be extended to humans. The adipose tissue of human subjects was found to constitutively express TNF-alpha and blood vessels of TNF-alpha fall after weight loss. But is this increase in TNF-alpha expression affecting insulin sensitivity? On binding of insulin to its receptor, tyrosine kinase activity in the cytoplasmic region of the receptor is activated. This receptor, tyrosine kinase, then phosphorylates both itself and some nearby proteins. The signaling cascade is initiated from the insulin receptor by the binding of adapter proteins such as GRB2 and IRS1 via their SH2 domains to the phosphorylated tyrosine sites on the receptor molecule. We now know that TNF-alpha signaling in adipocytes inhibits the autophosphorylation of tyrosine residues of the insulin receptor and instead induces phosphorylation of serine residues of both the insulin receptor and the IRS1 adapter. The serine phosphorylation inhibits any subsequent phosphorylation at tyrosine residues and thus the passage of signals from insulin to the interior of the fat cell is prevented. Recently, the interleukin IL-6 also has been shown to inhibit insulin signal transduction in hepatocytes through a similar mechanism. The decrease in effectiveness of insulin signaling then becomes a self-reinforcing problem as insulin signaling itself is anti-inflammatory, and so any decrease in the insulin signal can give rise to inflammatory side effects. However, TNF-alpha and interleukin-6 are not the only cytokines implicated in the etiology of type 2 diabetes. With the discovery and characterization of the pro-inflammatory cytokine family represented by IL-17, interest has risen in the relationship between members of this family, obesity, and the control of fat cell metabolism. Since IL-6 is implicated in the differentiation of T lymphocytes to secrete IL-17, obesity and its associated inflammation tend to predispose an individual to secrete IL-17. However, again we found ourselves in a positive feedback loop as IL-17 acts on monocytes to induce the further secretion of IL-6, thus ensuring the maintenance of an inflammatory state. It is therefore clear that cytokine signaling plays a profound role in a disease that is emblematic of our time and which is predicted to afflict close to 40% of the US population by the middle of the next decade.